All right, I think we can start. Good afternoon or good morning, and welcome to our February webinar of the International Society of Bone Morphometry. At the ISBM, we treat about all aspects of quantitative assessment of bone across scales, and becoming a member gives the opportunity to be part of leadership committees at different career stages, to benefit from collaborations and discussions with experts in the bone morphometry field. And it gives also the opportunity to be part of working groups. And we have currently 4 working groups running. You can see them on the slides. We have HRPQCT, Advancing Protocols for Clinical Research, Spatial Transcriptomics in skeletal tissues, Imaging and Analysis of Nerves in Bone, and Bone Micro-Ultrastructure Assessment Preclinical Models. I am Mathilde Palmier, and I am co-hosting this webinar with Annika Shimonti, and we are part of the Early Career Investigator Committee of the ISBM, and we are organizing this webinar series. Our next webinar will be end of March, and it will be about women in science, how to build a career. So we have 3 panelists and 2 moderators answering the panelists will answer the moderator questions, and the audience will also have some time to ask questions. So to receive the registration, to be aware of the registration modalities, please stay tuned, look at your emails, but also follow our LinkedIn page, and you will, which is regularly updated. It will also be the occasion to learn about our ISBM 2026 meeting, which will take place in London in July. And if you want to submit an abstract, hurry up, because the submission deadline is the 1st of March. Lastly, I would like to thank our corporate partner, AstraZeneca, for their support for this ISBM webinar series. And now it's time to welcome Dr. Rémy Gauthier, who is an associate researcher at the CNRS, a French National Center for Scientific Research. He's working in the MatéIS laboratory in the Material Science Department of INSA de Lyon in France. And he's working on bone biomechanics to improve bone fracture risk prediction and bone regeneration strategies. And he just got awarded with a grant from the French National Research Agency on the project Numerical and Experimental Modeling of Bone Mechanobiology. He also investigates osteocyte mechanical stimulation within the lacunocanalicular network in order to better understand bone remodeling and regenerative mechanisms. And today he will talk about analysis of human bone porous network by X-ray micro and Nano-CT. Welcome, Dr. Gauthier.
Thank you very much for this introduction. So I will just share my screen. Okay, so is it okay?
Yes,
you can see my PowerPoint. Okay, perfect. So thank you again for the introduction and for inviting me to present this work. So I'm very glad to present this because this is a work that has begun a long time ago when I was in PhD, actually, and we are still working on the data because we obtain very nice data from synchrotron radiation-based tomography. And so the other name that you can see here, Hélène Follet, David Mitton, and François Perrin, were my PhD supervisors. So yes, I will talk about human porous network analyzed by micro and nano computed tomography, and the context is the bone remodeling. So you may probably know what bone remodeling is, but I think it is important to just talk about it a bit to put my research in context. So bone is a mechanical material. As every mechanical material, when it is solicited mechanically, there is a damage formation. Here it is micro cracks are formed due to the mechanical loading. But for the bone to keep ensuring its role in our body, this micro crack needs to be repaired. And this is why biological mechanisms occur. So these biological mechanisms is the resorption of the damaged area by the osteoclast, the formation of a new bone matrix, and its mineralization by the osteoblasts. And all this is in fact identified. This damage area is identified because in fact when the bone is mechanically loading, the fact that there is micro cracks within the matrix means that the matrix will have a different mechanical behavior. And this is this mechanical behavior that acts as a signal for the biological actors that are around here to say, okay, we need to repair this matrix. And actually, the biological actor that we will focus on are the osteocytes, which are the cells that are deeply trapped within the mineralized matrix that can really act as a mechanical sensor and can locally feel their strain distribution really close deep within the mineralized matrix. So most of my work today on bone tissue are focused really on osteocytes, and mainly on trying to understand how osteocytes are stimulated mechanically in bone architecture. So the consensus is that osteocytes are mostly stimulated through the fluid that is flowing within the lacunocanalicular network. So the bone matrix is filled with a physiological fluid that is going in and out this porous network. And when the bone matrix is subjected to a mechanical loading, this porosity will change their dimensions. And with this dimensions change, there will be the formation of pressure gradients, and the pressure gradient will promote the fluid flow within this network. And it is in fact the shift in the flow of the fluid or in the pressure of the fluid within this network that is believed to be sensed by the osteocyte and then converted into some biological mediators. So the porous network is very important because the fluid is in this porous network, and it is the change of porous dimension that induce a change in the fluid behavior. Sorry. So the bone porous network is quite complex, as in many biological material, everything is multiscale. There is different scale that have to be considered. So if we look at the osteocyte, we can see that if we zoom out, we can see that the osteocytes are connected all together through a dense network of canaliculi. At a larger scale, even, we can see that there is a lot of osteocyte lacunar within the bone tissue. There is also a complex vascular network, and even if we go at the very big scale, we can see that there is a different type of vascular porosity between the trabecular and the cortical bone. And even if we talk about the complete organ, there is also the geometry that will have an influence on how the porous network is deformed during bone mechanical loading. And regarding what has been done in the literature, mostly using a numerical simulation, we can see that at every scale of this porous network, the morphology of the network has an influence on how the lacunocanalicular network is undergoing a fluid pressure shift or fluid velocity shift. For example, in Gauthier, they found that the size of the vascular canals may have an influence on the pressure and the velocity in the lacunocanalicular network. At the lower scale, it was shown that the shape of the osteocyte lacunae can have an influence on how the fluid will circulate within the canaliculi. And even at the lower scale, at the canaliculi, it has been shown that the arrangement or the architecture of the canalicular network have an influence on how fluid behaves within this network. So yeah, at every scale of the porous architecture of bone, there is an influence of the morphology on how the osteocytes will be stimulated if we consider that the osteocytes are stimulated by the fluid circulation. So the objective of what I will show you today is to use X-ray based tomography to analyze this multiscale porous network and try to understand if there are differences, for example, between different bone or between different regions in the bone. So let's first begin with the aversion network, so the vascular network. So to do this, this was data that we obtained during my PhD, so it was in 2015. We were lucky to be able to go to the European synchrotron radiation facility in France, on line ID 19 for those who know SRF. For this campaign, we studied 8 femoral diaphyses, femoral necks, and reduced diaphyses from the same donor, and the 8 donors were 8 female from 50 to 91 years old. The voxel size, the size of the volume of interest, that we analyzed at this time was this one, with the voxel size of 0.7 micrometers. So here you can see just a 3D rendering of what we have after the reconstruction of the phase-based contrast tomography. On this kind of volume, we applied a median filter just to remove the noise, and then with just a Otsu threshold, that is an automatic threshold that can use to separate two distinct classes in terms of cray level. We were able, in fact, to segment only the vascular network. So here, what you see in gray is not the bone; it's really the porosity, the vascular porosity. And here you can see just an example from the same donor, where you have the vascular porous network of the femoral diaphysis, the femoral neck, and the radius diaphysis. In terms of quantification, here I just show you the volume fraction of this canal within the total volume of the sample, and also the diameter of this canal. So the diameter is measured using an algorithm that, in fact, make a small sphere travel within the porous network, and the diameter is considered as the biggest sphere that can be fitted in the porosity. So here are the value. So these are value for cortical bone that are quite standard, below 10% of porosity. In terms of diameter, we can also see that we are quite standard in terms of what we can find in the literature. But what is interesting is the difference between the different anatomical sites. So here it means that for a same donor, the volume fraction of vascular porosity within the radius is significantly smaller than from the femoral diaphysis and the femoral neck. And if we consider the diameter, the radius diaphysis has smaller pore canals compared to the femoral neck. And if we just remember of the publication from Goulet and colleagues that say that the diameter of the vascular canal can have an influence on how the fluid behave in the lacunocanalicular network. This is interesting to see that for even the same donor, there is a difference between these 3 anatomical sites that may be obviously be explained, or we can make the hypothesis that, in fact, these 3 regions are not loading the same way during life, so the radius is much less loaded than the femoral neck and the femoral diaphysis, and that may explain why these morphological parameters are significantly different. So, okay, for the vascular porosity, let's go down a bit and try to investigate the osteocytes lacunae. So, in fact, we use the same sample. So the sample for 8 femoral diaphysis, femoral neck, and radius diaphysis for the 8 female donor. So this one, to segment the osteocytes lacunae, we use the hysteresis thresholding that allow to define a first threshold that is in the center of the lacunae, and then a second threshold that is used to tune the porosity to the border of the lacunae, the border of the lacunae and the bone, that is enhanced using a phase contrast-based microtomography. And then we did what we call a connected component analysis, meaning that you used an algorithm to distinguish every osteocyte lacunae between them. And so we are able to investigate and calculate some morphological parameter on each of these lacunae and then make some averages, for example. Here you can see just the 3D reconstruction of the volume that were used. So we use smaller volume for the lacunae compared to the vascular porosity, because there is a large number of lacunae, and decreasing the volume allowed us to decrease the computation time. To make some morphological parameter investigation, we considered that the osteocyte lacunae were in fact ellipsoid, and on this ellipsoid we were able to calculate, for example, the 3 different lengths of the 3 different axes. Here you can see the 3D rendering of the porosity. Again, what we see here are in fact, the wall of the different porosities. So we can easily recognize the vascular porosity, which are the large canals. In yellow, you have the lacunae, so the porosity that we are interested in. And also in red, you have the microcracks. So there is microcracks during life. So it is normal to investigate microcracks within bone samples. And also there is a risk that during the sample preparation, we also induce microcracks. So to distinguish microcracks from osteocyte lacunae, we applied a geometrical criteria. Basically, microcracks are planar defect and osteocyte lacunae are ellipsoid. So we are able, based on some geometrical criteria, to distinguish these two populations. In terms of parameter here, I just show you the osteocyte lacunae aspect ratio, so the main axis and the second axis, and the lacunar density, meaning the number of lacunae per unit volume of bone. So here you have the value. So in the literature, there is more and more data on lacunae, but at that time there was not so much data on lacunar morphology, but this is quite also classic values for lacunar morphological parameters. And again, what is interesting is the difference between the different anatomical sites. So we've seen that there were a difference in terms of vascular porosity, but again, here for the lacunar morphology, we have a difference in if we compare the radius, that is the non-loaded bone, compared to the femoral diaphysis and the femoral neck. And to be more precise, the lacunae from the radius are much more elongated. Well, more elongated, significantly more elongated compared to the lacunae that are in the femoral diaphysis and femoral neck. And this is interesting because, again, if we remember that Lay and colleagues did see that the size and the expansion of lacunae can have an influence on fluid flow within the canalicular network. Regarding these osteocyte lacunae, we also wanted to make a more local analysis. So we compare different bone from the same donor, but now what is the pattern within one bone? Because we know that in one bone we have some important features that are the osteons, and we wanted to compare, in fact, what the lacunae that are in the interstitial tissue to those that are in the osteons. And so here again, I just show you the same parameter, and we see also a significantly different morphological parameter. So for the first one, it is a very slight difference, but still it is very significantly different in the manner that, in fact, there is no if we look at the distribution of the lacunae within the osteon and the lacunae within the interstitial tissue, there is no range that the two distribution are. I don't remember the name, are in the same distribution. So the two distributions are very separated, very distinguished. And in terms of density, we can see that in the osteon, there is a higher density, a higher number of lacunae compared to the bone volume. To do an even more local analysis, we focused on the osteons, and we tried to understand if there was an evolution of the morphological parameters of the lacunae if we consider the distance from the central vascular canals to the cementlide, which is the frontier between the osteon and the interstitial tissue. Sorry. And here you can see the pattern. So this is the average pattern over all the osteon of all the donors. But if we take each donor, the pattern is really similar. You can see it on the paper if you want. But we can see that there is a specific pattern. For example, if we are looking the curve on the top, it means that the density of lacunae is higher, close to the cement line. And what can be hypothesized here is that close to the cement line, this is where the first new bone is formed, and maybe at this beginning of new bone formation, there is a higher density of osteoblasts that get trapped within the bone matrix, compared that with time and with the modeling of these new osteons, maybe less osteoblasts are embedded. regarding the aspect ratio, it is difficult to say if this is something that happened at the time of the osteocytes were embedded, or if this is something that evolved with time, because we know that osteocyte can remodel that perilacular matrix, so they can theoretically modify a bit their shape, and this would be interesting, for example, to investigate whether or not the osteocytes are able to tune their shape in order to promote fluid diffusion, fluid circulation, sorry, depending on the location on the osteon. For example, it will be of interest. So here are the results regarding the osteocytes lacunae, sorry. And now we can go down at the last scale that we investigate within this project, that are the canaliculi. So for this canaliculi, it is another protocol. We went on a double line of the ESRF, IDSSB. The voxel size here was 100 nanometer, and the volume were, of course, smaller. And the population were not the same, was not the same. So the population here are 27 femoral diaphyses from 16 female donors and 12 male donors. So here you can see just a slice of what we can obtain after the acquisition and the reconstruction of the data. So you can see clearly the vascular canal, the osteocyte lacunae, and we can detect some small canal that are a bit dispersed within the matrix, and these are the canaliculi. And to enhance their observation and allow their segmentation, we apply these different algorithms. So first, an enhancement of the features that are like vessels, so using the Sato's algorithm. Again, an hysteresis threshold, and then a variational region growing that allow to connect the canaliculi that were cut if they were close enough. Because we made the hypothesis that these canaliculi were connected, but the resolution that did not allow to observe them. So we applied this variational region growing to just reconnect the lost connection. So here you can find, you can just see an example of what we get after the segmentation. So in blue-green, you have the canaliculi, and in yellow, you have whether the osteocyte lacunae or the central vascular canals. And here is the focus on one osteons. So we can see, first qualitative assessment here is that there is a big difference between what is in the osteon and what is in the interstitial tissue. The interstitial tissue seems very less dense in terms of canaliculi network, and we can even see some lacunae that are not connected to any canaliculi. So this doesn't mean that there is no canaliculi, but still with this resolution we were not able to see them. In terms of quantitative parameter, here I show you the canalicular diameter and the canalicular volume fraction over the bone volume. So for the canalicular diameter, there were no difference between the female and male population, and actually, if we here can see a slight difference between the female and male donor for the canalicular volume fraction, it was not significant. We can also have a closer look on the osteocytes lacunae, really around one osteocytes lacunae, to maybe measure the number of canaliculi that are connected to a lacunae. So we can see that there is a large number of canaliculi that are connected to the lacunae. Here again, we can see a slight difference between female and male, but no significant differences. And also we managed to follow the evolution of the canalicular number, for example, or any canalicular morphological parameter. But here we focus on the canalicular number, depending on the distance to one osteocytes lacunae. And so here you can see that above you have the canalicular number that is increasing with the distance to the osteocytes lacunae, meaning that there is a ramification of these networks with the distance to the lacunae. But if we divide this number of canaliculi to the sphere that is englobing the osteocytes lacunae, meaning the sphere that with the radius that is the distance to the lacunae, we can see that in fact the density of canaliculi, decrease with the distance to the osteocytes lacunae. In addition, here I just wanted to show this because I think it's interesting. Here we can see just the osteocytes lacunae its surrounding canicular network. And here, what can we see is the cement line. And what is interesting, I think, is that we can see that there is a loss of the connection of the canaliculi through this cement line. So again, whether or not there are canaliculi, smaller canaliculi within this cement line, we cannot know now. But if there are canaliculi, they are too small to be observed with our resolution. And finally, we also wanted to measure some canalicular local properties. So this is the final part of my presentation. So here, what we wanted to do is to really consider one canicula in an isolated manner. And what we wanted to do is to propose a morphological parameter that is interesting in terms of fluid flow circulation. And in terms of fluid flow circulation, what we generally want to know is the shear stress that is applied on the canaliculi. So the shear stress is a function of the fluid velocity and the fluid viscosity. So for the fluid viscosity, we have to apply Darcy's law, for example, for a simple law of fluid flow. The fluid velocity is dependent on the hydraulic gradient and the bone intrinsic permeability. And in fact, this intrinsic permeability, we will be able to estimate it, at least based on some morphological parameter. And so we can use our data to propose a bone intrinsic permeability that can be used to estimate the fluid shear stress. So for that, we used permeability that is calculated using the Cosny relation. That is, in fact, a permeability used for porosity that looks like capillaries. And here in this relation, you have the pore volume fraction, the capillary diameter, and the tortuosity, which is in fact the actual length and the straight length. So how much the canalicula is tortuous. To analyze distantly each canalicula, we define a mesh. So from the segmentation, we calculated the distance map, and from the distance map, we were able to apply the watershed algorithm that, in fact, define a mesh based on the edge of the distance map. And in fact, so here you have a view in 3D. And in fact, what we were trying to do here is to define a mesh for which each edge of the cells is at equal distance from any canalicula. So in each of these cells, there is one canalicula, and in each edge of the cells, you have the same distance from any canalicula. And in each of these elements, in fact, we were able to apply this formulation. So here you can see the results in terms of 3D rendering. So we have a permeability that range from 0.2 10⁻⁷ to 10 10⁻17, square meter. So if we the mean and the median are around 10⁻10⁻18 square meter. And if we compare to what can be found in the literature, this is actually a bit small. But in fact, this can be explained by the fact that we only consider a permeability that is associated with the morphology of the canaliculi, and we don't consider what is inside the canaliculi during in vivo fluid flow circulation with the cellular and the pericellular matrices that have an influence on this permeability. Here, I just wanted also to show that there is a slight trend of decreased permeability with age. So actually, the correlation was significant. So the law is not strong, but there is this trend. And this would maybe mean that with the lower permeability, there is an altered remodeling capacity. But interestingly, what we have also found is that if we compare the female and male population, we have a decreased permeability in male, while in fact, usually we said that with old age, female will have a lower bone remodeling capacity. So this hypothesis is really questionable, and further investigation have to be done. We can also, as just for the canalicular network, we can do some local analysis close to the osteocyte lacuna. And here what we see is that the permeability is decreasing with the distance to the lacuna, which in fact is quite in accordance with the fact that we saw later with the decreased canalicular density with the distance to the lacuna. finally, I think this is also interesting. So here I show you the relation between the permeability and another parameter that is the canalicular linear density, which is in fact the length of the canalicular network divided by the bone volume. And here we can see that there is a really strong correlation between them. And this is interesting because the canalicular density is a parameter that can be obtained through confocal microscopy. So with really heavy image processing, but still maybe confocal microscopy is less heavy in terms of acquisition time and protocol than synchrotron-based radiation tomography. So this is very interesting. This means that in fact with confocal microscopy we will be able to estimate the permeability of the network of the bone with the canalicular density. So here some conclusion. So the importance of the multiscale porous network, the fact that there is difference between different bones, probably due to the fact that these bones are not mechanically stimulated the same way. Even within one bone, there are differences between the osteobones, for example, and the interstitial tissue. And what we have seen also is that the LCM permeability is very heterogeneous, and this probably suggests that a closer, more precise permeability law have to be used in numerical model when we want to investigate the fluid flow and the fluid pressure within the lacunocanalicular network during bone loading. So this is actually what we will do in the future, use these data to implement them into some numerical modeling, and try to estimate some interesting parameter. And with this, I thank you very much for your attention. I don't know what was my time, what was the time I should have respected, but I hope I was not too long.
Thank you, Dr. Gauthier, and you are not too long. Don't worry.
Okay, perfect.
We are open for questions right now, and you can put them in the chat or ask them directly. I'll start because I think I may have missed it. When we were showing the micro cracks, did you see that there's a difference in lacunar density around the micro cracks or no?
Oh, no, we didn't check that. This is an interesting point. We did not show that. We only tried to understand the influence of lacunar morphological parameters with some mechanical parameter associated with fracture properties, so the toughness of the bone. And we did find some significant correlation, but we did not check locally around microcracks if there are some difference in terms of osteocytes lacunae morphological parameter. But this is a good question.
And then with the toughness, would you expect more lacunar density if the bone is more tough?
So I think I don't remember exactly the results, but instinctively I would say that if there are more osteocyte lacunae, there is more defect in the bone, and the defect is source of stress concentration that increase the risk of fracture. So I would say that with just in terms of material science, with more lacuna, there will be a higher risk of fracture. But maybe also with more lacuna, there will be more remodeling, so different different materials property. So this is not so obvious, in fact. But I don't remember what was the result at the end. I just remember that a morphological parameter that was collected with a decreased toughness, so a higher risk for crack propagation, was the curvature of the lacuna. So the smaller was the curvature of the lacuna in the bone, the lower was the toughness. So really this can suggest that with a lower radius of curvature, there is a higher risk of stress concentration, and with a higher risk of stress concentration, there is a higher risk of fracture and crack propagation.
Thanks. I'll let other people ask questions now.
Okay.
Please go ahead, Richard.
Hi, thanks, Rémy. Great presentation. What I was so the shape parameters of the lacuna within osteons compared to interstitial bones. I found quite an interesting profile that you showed for the stretch of the lacuna.
This one.
Exactly here. bottom right, this is, can you say, so all the osteons, you included all the data from all the different osteons?
Yeah, we didn't, for one donor, one sample, we did not distinguish different osteons. So that may induce some bias. But really, if we look at the pattern for every sample, it is almost exactly the same, but just the average is not the same, but it is the same pattern.
Yeah, I find it striking. I'm just thinking, but for interstitial bone, if it was formerly an osteon, sometimes we do not know of where the osteon was placed, and this would give us some indication of to reconstruct earlier osteons. if this is
maybe if we look at if we take just some local subvolume and try to travel it around the bone, maybe we can see some interesting pattern. This is an interesting question too, actually.
Yeah.
But I don't know if during, because as I said, there is a possibility of perilacunar remodeling, and as soon maybe as the bone becomes interstitial tissue, maybe there is this remodeling, and that can change the lacunar morphology.
Yes. May I ask a second question of if the lacunae are mineralized, you lose them, or you can you separate them?
Well, it depends on the amount of mineralization. Here, we really based on the segmentation was really based on gray level. So if the lacuna is mineralized, the gray level will not be the same as for porosity. So for fully mineralized, we can see them actually in the data, but we did not consider them in the calculations.
Yes, I think this is also an interesting topic of how frequent they are.
Sure, we can, but they are not so easy to automatically segment because they are really close in terms of gray level. They are really close to the surrounding bone, so it's not so easy to segment them. So we have a lot of data. The volume are quite big, so we need some hands to segment this mineralized lacunae. But yes, it could be really interesting.
Yeah, thank you.
Thank you.
We have a question in the chat from Johannes Krug. I'm sorry if I butchered your name. Thanks for this nice overview and the great imaging. What is your take on the more recently described nanoporosity or nanochannels seen with FIB-SEM? Are you also looking into these length scales?
So there are a lot of work on, well, a lot. There are some work on these microchannels that are really interesting, but we were not able to resolve them with this nano computed tomography. But here is some FIB-SEM image of them. We just wanted to see if we were able to segment that. So we are able to segment that, and actually it seems very dense. The question is, are there really porosity, or are there some collagen that is loose during the sample preparation? So there is a lot of question about that. We are not focused on this nano channels. That is another network that can be interesting. But the lower scale I am working at is the canalicular scale.
Yeah, there's one from Benjamin Rutsky. Thank you very much for your talk. It was very interesting hearing about the differences between the osteons and the interstitial tissue. Has similar work been done in trabecular bone, or is the osteocyte network there less relevant or interesting due to the thinner, sparser nature of the trabeculae?
There is some work done on trabeculae. I don't remember. I'm not sure it was with synchrotron radiation, but with a lab scale tomography, I think there are some interesting work on iliac crest, for example, where they have done a huge work trying to analyze the morphological parameters of the osteocyte. So yes, there is some work about trabeculae, and I think they are as interesting as the one in cortical bone. And what can be interesting is to see along a whole section of bone to see if there is a gradient of morphological parameters, for example. And so, but we can hardly compare our results from this study with the other one on the iliac crest, because it's not the same population. I think in their study they are younger donor, so it's difficult to compare them directly.
Jonas, you can go ahead.
Hi, thank you for the previous answer. I just found a microphone. So I have another question in relation to this top view image that you showed of the Haversian canal with the canaliculi around it. And there you were seeing this disconnection, or like the canaliculi that definitely point towards the Haversian canal, but are somehow not connected to it.
Yeah, so this is a problem of the imaging methods that we used. If you look at here, so this is a problem. And it would be interesting to work on that, at least to investigate the number of connections to the vascular canal. This is a really interesting point. But if you can see here, you can see that around the canal there is some black area that is due to the phase retrieval of the X-ray on the detector. And here on this small region, we are not able to see all the canaliculi. So this is not something physiological. This is probably something that is associated with the imaging method and the imaging processing that we have used.
Okay, yeah, because I've seen similar stuff in confocal images, but there you also always have like some staining artifacts and so. So I was wondering.
Yeah, it is the same for the
explanation makes sense.
Yeah. Thank you.
Thanks.
Thank you. Naomi, you can go ahead.
Hi. Really cool talk, exciting stuff. I wanted to just comment on the trabecular bone side of things. So our lab actually has completed an experiment at ESRF as well on ID16 on trabecular bone. So I'm working right now on all that analysis, but look forward to sharing that with you guys soon, and then kind of describing more about the trabecular bone, because we're definitely really interested in it. And comparing that also to how the confocal is working in comparison to synchrotron, which has been cool. In terms of the cement lines and then the resolution limit, have you had a look at the U et al. Data from the 30 nanometer versus 120 nanometer resolution? They mention quickly, it's your PhD supervisor's lab that published it, but they mentioned quickly that they got one image of interstitial bone. I was wondering if anyone looked further at that to see if you can actually see canaliculi. I'm not sure if you would know.
So you mean if we are able with the 30 nanometer?
Yeah.
So I have to check. I have to check on the data. The problem with the 30 nanometer based images are the size of the volume that is very, very small. And I was not there when they acquired the data, but I don't know if it was really easy to choose the region that was scanned. So if we are lucky, we have some region where we can find this cement line, but I'm not sure it is so easy. I think like in the 30 nanometer volume, they have like, I don't know, 10 lacunae, 10 osteocytes lacunae, so it's very, very few. So I'm not sure we are good, but I can check on the data if we are located close to, well, if we can manage to see a cement line, and then we can see if we can see the canaliculi, yes.
Yeah, that'd be really interesting. And then for the graph where you talk about permeability versus age, you're saying that the permeability goes down with age. Is that also just correlated to number of lacunae, number of canaliculi, etc., or is that actually age independent? Do you know?
What do you mean?
Like, is this just a product of the size of the lacunae, the size of the canaliculi, the number of them, or is this actually like a separate thing with age?
I think it is something with the size of the canaliculi, right?
Okay,
the morphological parameters of the canaliculi.
Yeah. Okay, makes sense.
Yeah.
Thank you.
I may have one question.
Please go ahead.
So you say that you couldn't, with the resolution that you are using, you cannot see the canaliculi in the interstitial bone. So which would mean that the canaliculi are smaller. So in terms of mechanical fluid flow shear stress on the osteocyte, what would be the interpretation?
So I don't know if they are canalically or not. We don't know. We cannot know with this resolution. Again, if we use the 30 nanometer based images, maybe we can try and see if we are looking more canalically in this interstitial tissue. But I don't know if they are no canalically or if they are smaller. What I know from the literature is that with time the canaliculi can mineralize itself because I think the interstitial tissue is maybe more rigid, so there is less pressure gradient, so there is less fluid circulation, and also if we add that to the diminished permeability at the cement lines, there is less flow circulation within the interstitial tissue, so there is less movement of the fluid and movement of the ion. So maybe there is a mineralization of the process. In terms of fluid flow induced by the compressive loading, I don't know. I think if a canal is smaller, a same load will induce a faster velocity of a higher pressure, so a higher velocity of the fluid. So I think this was your question.
Yeah, the thing is, so if the canaliculi are mineralized, then it means that it would be an empty lacuna, right? The osteocyte would not be there anymore. Or even if there is no mechanical constraint anymore, then the osteocyte might not be that
Probably
functional.
Yeah.
Or,
Yeah. And there will start to, as Richard was talking about earlier, then the lacunae will also mineralize itself because the osteocyte will die, and by dying it will release some mineralized vesicle, and there will be some mineralization of the lacuna.
Yeah, and at the same time, then if the osteocyte would receive more mechanical fluid flow shear stress at that specific in interstitial bone, then I was wondering what it would mean to bone biology. Like to the why would the osteocyte need to be more stimulated at that specific in that specific area of the bone?
Okay. Well, I don't know. I think also I'm not talking about osteocytes that are stimulated. I will talk about osteocytes that are not stimulated. I think that if they are not stimulated, they will die, and by dying, they will also send some mediator to call the remodeling of this region. So yeah, as soon as it is stimulated, maybe it says, okay, here we can bring some nutrients, some oxygen by the fluid, so it is okay. You have not to remodel this region. I don't know. It is a hypothesis. I'm not a biologist, so I'm not sure about what I'm saying. But as soon as the osteocyte will die, it will mean that this region is too far, for example, from vascular porosity, and so it has to be remodeled to bring back some blood and some other nutrients that are needed for the cells to live.
Yeah. And that's quite interesting. I have done some confocal imaging of the lacunocanalicular network, and even with the confocal, then sometimes we are not sure if there is a canaliculi or not, and then what it means to the bone biology. Thanks.
Yeah, sure. Confocal is really interesting for that, but as it is based on an interconnectivity of the network for the rhodamide to go through all the canalicular network, if there is no connection, there will be no fluorescent marker, and so we are not sure if it is because there is not the canalicula or because there is no connection. So this is also interesting, and I think there are some paper done by Aurélien Gourrier, who is working on different image-based analysis of this canalicular network that can provide some answers to the questions.
Thanks.
Any more questions from anyone? Well, thank you so much, Dr. Gauthier. This was really interesting and really cool.
Okay, thank you. Thanks to you. Thank you for your question. It was really interesting discussing with you. And if you want to contact me to ask more questions, don't hesitate. I'm really glad to speak about that.
And thank you everyone for participating, and stay tuned for our next webinar as well as our ISPM conference in London. Thank you.
Goodbye. Thank you.